Hi again. Thanks for watching these videos. Now we're starting to get into a little bit of the niche topics, so that means that if you're watching, you're really into this, so thanks for that. Thumbs up. I'm going to remove myself. So in this video I'm going to talk about using negative controls. And if you go through these different libraries, you can see down here that there could be information about any negative controls. Several of them have so-called non-targeting controls, so these are guides that are not supposed to bind anywhere in the genome. You search for them by writing neg control, and this one has 1,000 of them. If you don't want to have 1,000, you can limit to top 100, for example, and then you'll see that you're going to get 100 negative controls, right, that you get out like this. Brie and Brunelli has both have, again, 1,000 negative controls. Gecko also has negative controls. These are different between these, of course, or at least to a large extent, or I'm sure there's an overlap, but of course you could use negative controls from another library into your library. It doesn't matter, right? As long as you're using the right species, like, right, if you're using a human, if you're in a human setting, you want to use a human negative control, etc. The Gattinara and the Goda library, both of them have 500 non-targeting controls, but they also have this, what we call cut control, or safe targeting controls, and these have one cut site outside of any gene and can be used as a control for induced DNA damage response. And this is something you could think about, and you probably are when related to CRISPR, right? That you're not only inactivating a gene, you're also activating a DNA damage response, and that could in certain settings affect your cells, especially if you combine several guide RNAs, and if those guide RNAs have several off-target activities, then you start to accumulate and actually can have a reasonably strong activity via activation of p53. So these type of controls could be interesting to include in the library. They probably will more likely negatively impact yourself than the non-targeting control, as you have this p53 activation, this DNA damage response. Although keep in mind that many cell lines have mutated p53. So if you're working with standard cell lines, it's a good chance that they are mutated for p53. And then they don't necessarily have much of a DNA damage response, right? Although there could be subtleties in that. So that's how you use them. The VBC, at least the library that you download from this link, doesn't have any negative controls. I think they had negative controls when they do their screens. But here there's not, right? But so that's more or less everything about negative controls. You can use any library that has controls for your screen. You just need to download the controls and then sort of add it to your list. But why not using the same one that you get in your other spacers from? If you're not, just make sure that you do correct with mouse or human. Most of them have these negative controls. The Gattinara and Goda has this cut control. I think these are really good, and you limit to top to sort of limit the amount of controls you want to have. And you can see them. No, yeah, VBC doesn't have any. And you can see them like this, right? All right. Thank you again.